LAMARCKISM, concept, significance, and relevance in modern biology. The theory of Lamarckism, proposed by Jean-Baptiste Lamarck in the early 19th century, marks the first comprehensive and systematic attempt to explain the process of evolution. His work, especially elaborated in his 1809 book Philosophie zoologique, predates Darwin's theory by about 50 years and introduced the idea that organisms change over time due to interactions with their environment. Lamarck's theory is commonly referred to as the theory of inheritance of acquired characters. He postulated that organisms develop characteristics through use or disuse of organs in response to environmental needs, and these acquired traits are passed on to offspring. This was a groundbreaking concept at the time, as it implied a dynamic interaction between the organism and its environment in shaping evolutionary outcomes. The two most important principles of Lamarck's theory are: 1. Use and disuse. Organs that are used extensively become more developed and efficient, whereas those not used deteriorate over time. For example, Lamarck cited the elongated neck of the giraffe as a result of its ancestors stretching their necks to reach higher leaves. 2. Inheritance of acquired characters. Traits developed during an organism's lifetime due to environmental influence or behavior could be passed on to its progeny. Lamarck believed that the physical and behavioral changes in response to challenges could accumulate over generations, resulting in speciation. Lamarckism was initially influential but eventually rejected by most biologists due to the lack of empirical evidence and the failure to observe the transmission of acquired traits in controlled experiments. August Weismann's germplasm theory dealt a critical blow to Lamarckism. Weismann demonstrated, through his famous mouse tail cutting experiments, that modifications in somatic cells, like cutting tails, do not affect germ cells, thus acquired traits cannot be inherited. Despite its fall from favor, Lamarck's contribution to evolutionary thought remains significant. He was one of the first to propose that organisms are not fixed and that evolution occurs through natural processes. Modern evolutionary biology does not accept Lamarckism in its original form, but a neo-Lamarckian perspective has emerged with the advent of epigenetics. Epigenetic modifications, such as DNA methylation and histone modification, can be induced by environmental factors and, in some cases, passed to subsequent generations without altering the DNA sequence. This has led to a reevaluation of Lamarckian ideas in a modern molecular context. However, these epigenetic changes usually do not persist for many generations and do not result in long-term evolutionary change in the same way as mutations in germline DNA. In addition, Lamarck's emphasis on environmental influence on phenotypic plasticity is increasingly appreciated. Environmental stressors can shape organismal traits that enhance survival, which may later be stabilized by natural selection if genetic variations support those traits. Although Darwinian natural selection remains the core mechanism of evolution, Lamarck's ideas paved the way for integrating environmental responsiveness into evolutionary theory, which modern biology explores through developmental plasticity, gene regulation, and environmental genomics. Thus, while Lamarckism is not accepted as a standalone evolutionary mechanism, it is respected for its pioneering vision. The theory set the stage for subsequent models of evolution, encouraged empirical investigation into the inheritance of traits, and is philosophically and historically significant. For the CSIR-NET exam, understanding Lamarck's principles, his historical context, criticism, and modern reinterpretations through epigenetics is crucial, especially as questions often test comprehension of conceptual evolution history and integration with modern biology. Previous year questions from CSIR-NET. CSIR-NET June 2015. Which of the following best explains Lamarck's theory of evolution? A natural selection acting on random variation. B. Inheritance of acquired characters. C. Survival of the fittest. D. Genetic drift. Answer: B. Inheritance of acquired characters. CSIR-NET December 2013. The idea that organisms can pass on characteristics that they acquired during their lifetime to their offspring is known as Darwinism. B. Mendelian inheritance. C. Lamarckism. D. Epistasis. Answer: C. Lamarckism. CSIR-NET June 2017. Weismann's experiment involving the removal of tails in mice challenged Lamarck's concept by demonstrating acquired characters are heritable. B. Germline is unaffected by somatic changes. C. All mutations are beneficial. D. Evolution is goal-directed. Answer: B. Germline is unaffected by somatic changes. Let me know the next topic you want in this format. I can continue building your full CSIR-NET Life Sciences lecture set one by one. You said. Darwin concepts of variation. ChatGPT said. Here is your CSIR-NET JRF Life Sciences Advanced Level Lecture on Darwin. Concepts of variation, structured with full theoretical depth, integrated with previous year questions, and tailored for exam success. Darwin's theory and concepts of variation in evolutionary biology. Charles Darwin's theory of evolution by natural selection, as presented in On the Origin of Species, 1859, revolutionized biological sciences by providing a natural explanation for the diversity of life on Earth. At the core of his theory lies the concept of variation, which Darwin regarded as the raw material upon which natural selection acts. He observed that individuals within a population are not identical. Instead, they exhibit subtle differences in traits, referred to as variations. These variations, when heritable, could lead to differential survival and reproduction, shaping the evolutionary trajectory of species over generations. Darwin distinguished between two major types of variation: continuous variation and discontinuous variation. Continuous variations are small, incremental changes that occur among individuals and form a gradient, such as height or skin color in humans. These are usually polygenic traits influenced by multiple genes and environmental interactions. Discontinuous variations, on the other hand, are discrete and categorical, such as blood groups or flower color in peas, typically governed by a single gene with Mendelian inheritance. Darwin placed more emphasis on continuous variation, as he believed evolution proceeded through the accumulation of small, gradual changes. At the time of Darwin, the mechanisms underlying inheritance were unknown. He proposed a now defunct hypothesis called pangenesis, which suggested that all parts of the body produce tiny particles, gemmules, that migrate to the reproductive organs and influence heredity. While this idea was incorrect, Darwin correctly inferred that variation is heritable and that individuals with beneficial variations tend to survive longer and produce more offspring, a process he termed natural selection. The modern understanding of variation was only fully established after the rediscovery of Mendelian genetics and its fusion with Darwinian selection theory during the modern synthesis, 1930s-40s, where population genetics explained how variations arise and are inherited. The sources of variation include both genetic and environmental factors. Genetic variation arises from mutations, gene flow, genetic recombination during meiosis, crossing over and independent assortment, and chromosomal alterations. Mutation, especially, introduces new alleles into a population, while recombination reshuffles existing alleles to generate new combinations. Environmental factors contribute to phenotypic plasticity, where the same genotype may express different phenotypes under different conditions, though these are not heritable in the Darwinian sense. From a Darwinian perspective, variation must meet three conditions to be evolutionarily significant. 1. It must be phenotypically expressed, affecting traits that influence fitness. 2. It must be heritable, ensuring that offspring can inherit the trait. And 3. It must affect reproductive success, with individuals bearing advantageous traits producing more viable offspring. Traits that do not impact survival or reproduction may remain neutral and subject to drift rather than selection. In the post-Darwinian framework, variation is also linked with adaptive evolution, where alleles that confer fitness advantages increase in frequency and elevation over time. Directional selection favors one extreme phenotype, stabilizing selection favors the average, and disruptive selection favors both extremes over the mean. Each of these modes of selection operates on heritable variation, and their dynamics are shaped by environmental context and population structure. Understanding the genetic basis of variation has also allowed scientists to study phenomena such as balancing selection, where multiple alleles are maintained in the population due to heterozygote advantage or frequency-dependent selection. These concepts are especially relevant in disease resistance, e.g., sickle cell trait and malaria resistance, post-pathogen coevolution, and the maintenance of biodiversity. For CSIR-NET preparation, it is essential to link Darwin's qualitative observations on variation with modern quantitative genetic principles. Questions in the exam often target the types of variation, mechanisms producing variation, and how selection acts upon them, including theoretical models and applied examples in population genetics, ecology, and evolution. Previous year questions from CSIR-NET. CSIR-NET December 2016. Darwin considered which of the following is the raw material for evolution? A. Mutation. B. B genetic drift, C variation, D speciation. Answer, C variation. CSIR net June 2013. Which one of the following is not a source of genetic variation in a population? A mutation. B recombination, C natural selection, D gene flow. Answer, C natural selection. Explanation, natural selection acts on variation but does not generate it. CSIR net June 2018. Which of the following best describes discontinuous variation? A variation along a continuous gradient, B variation caused only by environment, C distinct phenotypic classes with no intermediates, D gradual and cumulative changes in phenotype. Answer, C distinct phenotypic classes with no intermediates. Let the next topic you like. I recommend continuing with Darwinian natural selection and adaptation, or we can move to any other topic in evolution, genetics, or cell biology as you prefer. You said, adaptation, ChatGPT said. Here is your CSIR net JRF Life Sciences Maximum Detail Lecture on Adaptation, crafted with exam precision, structured in dense paragraph format with no space wasted, and including previous year CSIR questions and answers for solid exam grounding. Adaptation, concept, mechanisms, and evolutionary significance. Adaptation, in evolutionary biology, refers to the process by which organisms become better suited to survive and reproduce in their specific environments through evolutionary changes. It is both a process and a state. As a process, it describes the evolutionary mechanisms, primarily natural selection, by which populations acquire traits that enhance survival and reproduction. As a state, it refers to the actual traits that increase fitness. Adaptations arise over many generations through the differential survival and reproduction of individuals with heritable traits that provide a selective advantage under specific environmental conditions. Charles Darwin placed adaptation at the center of his theory of natural selection. He proposed that individuals with traits better suited to their environment have a higher chance of survival and reproduction, leading to the gradual accumulation of these traits in the population. The modern understanding of adaptation integrates genetic variation, phenotypic expression, and selection pressures. Genetic variation, arising from mutation, recombination, and gene flow, provides the raw material upon which natural selection acts. When a particular phenotype confers a survival or reproductive advantage, alleles contributing to that phenotype increase in frequency, a process described quantitatively in population genetics using fitness coefficients and selection gradients. Adaptations can be classified based on function and origin. Structural adaptations involve morphological features such as the streamlined body of fish or the thick fur of Arctic mammals. Physiological adaptations include biochemical and metabolic modifications, such as antifreeze proteins in polar fish or C4 photosynthesis in plants living in high-temperature environments. Behavioral adaptations involve changes in activity patterns, foraging behavior, or reproductive strategies, such as migration, hibernation, or social cooperation. Not all traits are adaptive. Some may be neutral or even maladaptive due to genetic drift, pleiotropy, linkage disequilibrium, or evolutionary lag. A distinction must also be made between adaptation and acclimatization. While adaptations are genetic and heritable, acclimatization refers to reversible, non-heritable physiological adjustments that organisms make in response to environmental changes, such as increased red blood cell production at high altitudes in humans. At the molecular level, adaptation involves changes in allele frequencies over time, often driven by directional or stabilizing selection. For example, antibiotic resistance in bacteria is a classic case of rapid adaptation under strong selection pressure. In such populations, a mutation-confirmed resistance provides a significant fitness advantage, leading to a swift increase in the frequency of that allele. Similarly, in host parasite systems, adaptations occur in an arms race manner, where evolutionary changes in one organism drive reciprocal changes in the other, a concept known as coevolution. Adaptations can also result from convergent evolution, where unrelated species independently evolve similar traits due to similar environmental pressures. For example, the wings of bats and birds are structurally different but functionally similar adaptations for flight. Another example is the evolution of antifreeze proteins in Arctic and Antarctic fish, which evolved independently but serve the same function. Adaptations are constrained by historical, developmental, and genetic factors. Not all phenotypic changes are optimal, and evolution works with pre-existing structures and genes. This concept, known as historical contingency, means that organisms evolve the best solution available within the limits imposed by their ancestry, not necessarily the perfect one. In evolutionary theory, understanding adaptation is crucial for explaining biodiversity, niche specialization, speciation, and organismal design. In applied biology, studying adaptations aids in conservation biology, agriculture e.g., crop resilience medicine e.g., understanding pathogen evolution and climate change responses. From a CSIR net perspective, questions may focus on recognizing adaptive traits, distinguishing them from acclimatization or phenotypic plasticity, identifying selection pressures, and analyzing evolutionary patterns through examples and experimental data. Previous year questions from CSIR net. CSIR net December 2016. Which of the following best defines adaptation in evolutionary terms? A temporary physiological change. B heritable trait that increases fitness in a given environment. C learned behavior in animals. D response to seasonal changes. Answer, B heritable trait that increases fitness in a given environment. CSIR net June 2014. Antibiotic resistance in bacteria is an example of acclimatization. B genetic drift. C adaptation through natural selection. D artificial selection. Answer, C adaptation through natural selection. CSIR net December 2019. Which one of the following statements is correct with respect to adaptation and acclimation? A both are genetically heritable. B adaptation is reversible. Acclimatization is not. C adaptation is heritable. Acclimatization is not. D acclimatization occurs over generations. Answer, C adaptation is heritable. Acclimatization is not. Let me know the next topic. You can continue with evolutionary themes like natural selection, speciation, or go into genetics, cell biology, or any other core area of CSIR net life sciences. You said, struggle, ChatGPT said. Here is your maximum detail CSIR net JRF life sciences lecture on struggle for existence, written in dense paragraph format, with zero space wasted, tailored specifically for CSIR level preparation from basics to advanced concepts, and includes selected previous year CSIR questions with answers. Struggle for existence, Darwinian foundation and modern understanding. The concept of struggle for existence is a foundational pillar of Darwin's theory of evolution by natural selection. It refers to the competition among organisms, both within and between species, for limited resources such as food, space, mates, and other environmental necessities. This struggle is not merely physical conflict, but encompasses all biological pressures that limit an organism's reproductive success. Darwin was influenced by the economist Thomas Malthus, who argued that populations grow geometrically while resources increase arithmetically, inevitably leading to competition. Darwin extended this to the natural world, proposing that more offspring are produced than can survive, and thus, only those best suited to the environment survive and pass on their traits, a process we now understand as natural selection. There are three types of struggle: intraspecific, interspecific, and environmental. Intraspecific struggle occurs within members of the same species and is the most intense because the competitors share identical ecological needs. For example, deer in a forest compete for the same type of foliage and mating opportunities. Interspecific struggle occurs between different species competing for overlapping resources. For instance, lions and hyenas compete for herbivorous prey. Environmental struggle refers to challenges posed by abiotic factors like drought, temperature extremes, floods, or disease. These factors can impose strong selective pressures independent of direct biotic competition. The struggle for existence acts as a filtering mechanism in natural selection, determining which phenotypes confer survival advantage in a given environment. This selective pressure leads to the differential survival and reproduction of individuals with favorable traits. The heritable traits that confer even a slight edge in this struggle accumulate in the population over generations, leading to adaptation and possibly speciation over evolutionary timescales. In modern evolutionary biology, this concept is deeply integrated with population genetics, where the frequency of alleles that enhance fitness increases due to the differential reproductive success resulting from this struggle. From a molecular and ecological standpoint, the struggle for existence is evident in competitive exclusion, resource partitioning, and niche differentiation. The competitive exclusion principle states that two species competing for the exact resources cannot coexist indefinitely. One will outcompete the other unless they evolve to utilize different niches, a process often facilitated by natural selection. This is observed in ecological field experiments and theoretical models like lockable Terra competition equations, where species coexist only when they use different portions of the resource space. Moreover, the struggle for existence drives evolutionary arms races in predator-prey dynamics, host-pathogen interactions, and plant-herbivore relationships. For example, plants may evolve toxic compounds to deter herbivores, while herbivores evolve mechanisms to detoxify those compounds. Such interactions illustrate how the struggle for survival can drive complex coevolutionary adaptations. Importantly, the struggle is not necessarily visible as direct aggression or competition. For instance, in microbial populations, struggle manifests through faster replication rates, biofilm formation, secretion of inhibitory compounds, and resistance mechanisms. In sessile organisms like plants, struggle includes competition for sunlight, root space, or pollinators. The cumulative effect of all these biotic and abiotic interactions results in natural selection shaping population structure, adaptive radiation, and niche specialization. From an exam perspective, the struggle for existence is often assessed not only as a historical concept from Darwinian theory but also in its modern integrated form, connected to ecological theory, fitness landscapes, and population dynamics. Students are expected to apply this concept to real-world examples, experimental scenarios, and data interpretation questions that test the understanding of evolutionary pressures and selection mechanisms. Previous year questions from CSIRNet. CSIRNet, December 2013. Which of the following is an example of interspecific competition? A lions and hyenas competing for prey. Two species of birds competing for nesting sites. C. Male deer fighting for access to females. D. Plants and herbivores in an ecosystem. Answer. C. Male deer fighting for access to females. CSIRNet, June 2015. According to Darwin, the struggle for existence is most intense between a closely related species. B. Distantly related species. C. Individuals of different trophic levels. D. Individuals of the same species. Answer. D. Individuals of the same species. CSIRNet, December 2017. Competitive exclusion results from which of the following? A shared evolutionary history. B. Sympatric speciation. C. Similar ecological requirements and overlapping niches. D. Mutualism between two species. Answer. C. Similar ecological requirements and overlapping niches. Let me know your next topic: speciation, types of selection, genetic drift, or move into cell biology, biochemistry, or molecular biology. I'll continue with maximum quality and detail. You said, Fitness ChatGPT said. Here is your CSIRNet JRF Life Sciences Ultra Detailed Lecture on Fitness, crafted exactly as per your instructions. Dense paragraph format, no tables or space wastage, from base to advanced level, and incorporating CSIRNet previous year questions with answers in years. Fitness in evolutionary biology, concept, types, and molecular interpretations. In evolutionary biology, fitness refers to the reproductive success of an organism relative to others in the population. It is not merely about survival but about the ability to survive, reproduce, and pass on genes to the next generation. The term was popularized through Darwinian theory, though Charles Darwin himself used the phrase survival of the fittest metaphorically. In modern evolutionary synthesis, fitness is quantified as the average number of offspring contributed by an individual to the next generation's gene pool, often relative to the most successful genotype. Thus, it is a relative measure, not absolute. Fitness is fundamentally a genotype-dependent trait because different genotypes can lead to variations in reproductive success. For instance, in a population of beetles, if a certain color morph avoids predation better and reproduces more successfully, the allele responsible for that coloration has higher fitness. This difference in reproductive output leads to natural selection, where alleles associated with higher fitness increase in frequency over generations. Hence, fitness is the driving engine of evolution through selection. There are different types of fitness depending on context. Absolute fitness W refers to the average number of offspring produced by individuals with a particular genotype. Relative fitness W is the fitness of a genotype compared to the genotype with the highest reproductive success in the population, which is arbitrarily assigned a fitness value of 1. For example, if genotype A produces an average of 4 offspring and genotype B produces 2, then the absolute fitness of A is 4, and that of B is 2. But if we normalize it, relative fitness of A equals 1 and B equals 0.5. Darwinian fitness encompasses both survival and fecundity components, as it reflects the organism's contribution to the gene pool over time. Fitness can be influenced by many factors: survival until reproductive age, mating success, fertility, fecundity, and viability of offspring. These components together contribute to inclusive fitness, which also accounts for the reproductive success of genetically related individuals. The concept of inclusive fitness is central to the theory of kin selection, proposed by W.D. Hamilton, where organisms may increase their genetic representation in future generations by helping close relatives reproduce, even at a cost to their own direct reproduction. This explains altruistic behaviors in eusocial species like bees, where sterile workers help weirder siblings. Another key dimension is context dependence of fitness. A genotype's fitness can change depending on environmental conditions or the frequency of other genotypes. This is seen in frequency-dependent selection, where the fitness of a phenotype depends on its frequency in the population. In negative frequency-dependent selection, rare genotypes have higher fitness, which helps maintain genetic diversity, while in positive frequency-dependent selection, common phenotypes are favored, potentially leading to fixation. Fitness landscapes, introduced by Sewell Wright, provide a metaphorical way to visualize how populations evolve towards peaks of higher fitness through mutation, genetic drift, and selection. In this landscape, genotypes are mapped onto a multidimensional space where height corresponds to reproductive success. Populations may get trapped on local fitness peaks unless significant genetic changes allow them to cross valleys and reach higher peaks, explaining constraints and potential in evolution. In molecular terms, fitness can also be linked to allele frequencies and selection coefficients S. If a genotype has a fitness disadvantage, it is assigned a selection coefficient S such that W equals 1S. For example, if a deleterious mutation reduces reproductive output by 10%, then S equals 0.1 and W equals 0.9. This mathematical framework is vital in population genetics to model allele frequency dynamics under various selection pressures. Importantly, fitness is not a property of an individual trait but of the entire phenotype in a given environment. A trait may be advantageous in one environment but neutral or harmful in another. This is the basis of adaptive trade-offs, where increased fitness in one trait may reduce fitness in another, e.g., increased size may improve survival but reduce agility or reproductive output. For CSIRNet, understanding fitness is crucial because it underpins natural selection, adaptation, population genetics, and evolutionary stability. It frequently appears in questions related to allele frequency changes, selection models, behavioral ecology, and molecular evolution. Previous year questions from CSIRNet. CSIRNet, June 2014. If genotype A produces an average of 4 offspring, A produces 3, and A produces 2, what is the relative fitness of genotype A? Answer. Relative fitness equals fitness of A. Fitness of fittest genotype equals 3/4 equals 0.75. CSIRNet, December 2017. Which of the following statements is true about fitness in evolution? A fitness is always about survival. B. Fitness includes only number of offspring. C. Fitness depends on environment and reproductive success. D. Fitness is an absolute property of a genotype. Answer. C. Fitness depends on environment and reproductive success. CSIRNet, June 2016. The selection coefficient S of a genotype is 0.2. What is its relative fitness W? Answer. W equals 1 S equals 1 to 0.2 equals 0.8. Let me know the next topic to continue your CSIR Life Sciences crash coaching. You can go deeper into selection types, speciation, population genetics, or shift to cellular and molecular biology as needed. You said, natural selection. ChatGPT said. Here is your highly detailed JRF level CSIR Net Life Sciences lecture on natural selection, written in dense paragraph form with no tables or wasted space, based on previous year CSIR questions, and structured from basic to advanced understanding. This is tailored to be your sole and final reference for mastering this topic for CSIR Net JRF. Natural selection, core mechanism of evolutionary change. Natural selection is a central mechanism of evolution first proposed by Charles Darwin and later integrated with Mendelian genetics during the modern evolutionary synthesis. It is defined as the differential survival and reproduction of individuals due to differences in phenotype. This process results in the increase in frequency of advantageous alleles and the decrease of deleterious ones in a population over generations. Natural selection is non-random, but it operates on variations that arise randomly through mutations, recombination, and genetic drift. The principle of natural selection rests on three core postulates: variation, heredity, and differentness. Individuals in a population vary in traits. Some of this variation is heritable, and individuals with traits better suited to the environment tend to survive and reproduce more successfully. There are several types of natural selection, each affecting the shape of the trait distribution in a population. In directional selection, one extreme phenotype is favored over others, leading to a shift in the population's trait distribution. This is common in response to environmental changes, such as antibiotic resistance in bacteria or industrial melanism in peppered moths. Stabilizing selection favors intermediate phenotypes and reduces variation by selecting both extremes. An example is human birth weight, where very low or very high weights are selected against. Disruptive selection, in contrast, favors individuals at both extremes of a trait and may lead to speciation, especially when combined with reproductive isolation. For instance, in a heterogeneous environment, two distinct beak sizes in finches may be favored for feeding on different seed types. In balancing selection, multiple alleles are actively maintained in the population through mechanisms such as heterozygote advantage, e.g., sickle cell allele conferring malaria resistance, or negative frequency dependent selection, rare phenotypes having higher fit. In balancing selection, multiple alleles are actively maintained in the population through mechanisms such as heterozygote advantage, e.g., sickle cell allele conferring malaria resistance, or negative frequency dependent selection, rare phenotypes having higher fitness. Selection can operate at multiple levels: gene, individual, kin, and group. While gene level selection highlights selfish genetic elements like transposons, individual selection explains most evolutionary outcomes. Kin selection, a form of inclusive fitness, explains altruistic behaviors in social organisms, where individuals sacrifice direct reproduction to help close relatives. Group selection, although controversial, refers to natural selection acting at the level of entire groups, especially in cases where cooperative groups outcompete selfish ones. The genetic consequences of natural selection are measurable through changes in allele frequencies over time. The selection coefficient S quantifies the disadvantage of a genotype relative to the fittest genotype, which has W equals 1. For example, if genotype A has a fitness of 0.8 compared to genotype A with fitness 1.0, then S equals 0.2. These parameters are crucial in Hardy-Weinberg-based models used in population genetics. In polygenic traits, natural selection shapes the adaptive landscape, influencing the movement of populations toward local or global fitness peaks, as visualized by Wright's fitness landscapes. Natural selection is context-dependent, meaning that the selective advantage of a trait is influenced by ecological, temporal, and population-based factors. For instance, in red queen dynamics, host-parasite coevolution drives continuous adaptation with no absolute gain in fitness, emphasizing the dynamic nature of selection. Additionally, sexual selection, a form of natural selection, acts on traits that affect mating success rather than survival. This includes intersexual selection, mate choice, and intersexual selection, male-male competition, explaining exaggerated traits like peacock feathers or deer antlers that may not confer survival advantage but enhance reproductive success. Experimental evidence for natural selection comes from long-term evolution experiments, LTEs, fossil records, comparative genomics, and real-time ecological studies. For example, Peter and Rosemary Grant's studies on Galapagos finches provided irrefutable proof of rapid evolutionary changes due to natural selection during droughts, where beak size evolution occurred within a few generations. Modern genomic tools allow mapping of selective sweeps, where regions of the genome show reduced variation due to positive selection. For CSIR Net JRF preparation, understanding natural selection is vital not just in theory but also in application. Questions often involve interpreting data on fitness, allele frequency changes, Hardy-Weinberg deviations, types of selection, and adaptive significance in ecological contexts. Candidates are expected to connect selection with evolutionary theory, population genetics, behavioral ecology, and molecular evolution. Previous year questions from CSIR Net Life Sciences. CSIR Net December 2016. Which of the following is an example of stabilizing selection? A emergence of antibiotic resistance B selection against very small and very large birth weights in humans. C divergence of beak sizes in finches. C increase in frequency of dark moths in polluted areas. Answer: B selection against very small and very large birth weights in humans. CSIR Net June 2015. Which of the following conditions is not essential for natural selection to occur? A heritable variation in traits. B unlimited reproductive potential. C equals survival of all genotypes. D differential reproductive success. B unlimited reproductive potential. C equals survival of all genotypes. D differential reproductive success. Answer: C equals survival of all genotypes. CSIR Net December 2018. In a population of insects, green coloration is advantageous in forest habitats. Over generations, green coloration becomes predominant. What type of selection is this? A directional selection. B stabilizing selection. C disruptive selection. D genetic drift. Answer: a directional selection. Let me know your next topic. I can continue with genetic drift, speciation, Hardy-Weinberg equilibrium, or any module from molecular biology, biochemistry, developmental biology, or methods in biology. I'll maintain the same high detail in CSIR focus. You said, Mendelism, ChatGPT said. Here is your CSIR Net JRF Life Sciences Ultra Detailed Lecture on Mendelism, structured exactly as you requested, dense paragraph format, maximum word coverage, no tables, highly exam-oriented, and includes 2 to 3 CSIR Net previous year questions with answers in years. This is crafted as a complete lecture from base to advanced concepts. Mendelism, principles, extensions, and genetic interpretation in modern biology. Mendelism refers to the foundational principles of heredity established by Gregor Johann Mendel through his pioneering experiments on Pisum sativum garden pea. His work, published in 1866 but rediscovered in 1900, laid the cornerstone of classical genetics. Mendel observed patterns in the inheritance of traits such as seed shape, flower color, and pod color across generations. From these observations, he deduced 2 key laws: the law of segregation and the law of independent assortment. These principles describe how discrete units of heredity, now known as genes, are transmitted from parents to offspring in predictable ways. The law of segregation states that each individual possesses 2 alleles for each gene, one inherited from each parent, and these alleles segregate independently during gamete formation such that each gamete receives only 1 allele. This law explains the 3:1 phenotypic ratio observed in a monohybrid cross between 2 heterozygotes. For instance, crossing 2 pea plants heterozygotes for seed color YY times YY produces a phenotypic ratio of 3 yellow, 1 green seeds in the F2 generation. This is due to the genotypic ratio of 1 YY, 2 YY, 1 YY, where both YY and YY yield yellow seeds due to dominance of the Y allele. The law of independent assortment asserts that alleles of different genes assort independently of each other during gamete formation. This was demonstrated using dihybrid crosses, where Mendel observed a 9:3:3:1 phenotypic ratio among F2 progeny when examining 2 traits simultaneously, e.g., seed color and seed shape. This law is valid only when the genes are located on different chromosomes or far apart on the same chromosome to avoid linkage. While Mendelian genetics laid the groundwork for understanding inheritance, modern extensions of Mendelism explain complex genetic phenomena that deviate from classical ratios. These include incomplete dominance, codominance, lethal alleles, multiple alleles, epistasis, pleiotropy, polygenic inheritance, and sex-linked traits. In incomplete dominance, the heterozygote displays an intermediate phenotype, e.g., red times white flowers producing pink in snapdragons. In codominance, both alleles are fully expressed, as in ABO blood group inheritance, where alleles E and IB are both expressed in heterozygotes IAIB. Lethal alleles can cause deviation from Mendelian ratios by resulting in the death of certain genotypes. For instance, a cross between two heterozygous yellow mice YY times YY yields a 2:1 phenotypic ratio because the YY genotype is lethal. Epistasis involves interactions between genes at different loci, where one gene masks or modifies the expression of another gene. An example is the recessive epistasis observed in Labrador retriever coat color, where the expression of coat pigment depends on both pigment-producing and pigment-depositing genes. These gene interactions often produce modified dihybrid ratios such as 9, 7, 12, 3, 1, or 9, 3, 4. The understanding of linkage and recombination refined the law of independent assortment, showing that genes located close together on the same chromosome do not assort independently due to linkage. However, recombination through crossing over during meiosis can break linkage and generate new combinations. The frequency of recombination is used to construct genetic linkage maps, where 1% recombination equals 1 centimorgan cm. Mendelism also applies to humans in studying single gene disorders, such as cystic fibrosis, autosomal recessive, Huntington's disease, autosomal dominant, and hemophilia X-linked recessive. Genetic pedigrees are analyzed using Mendelian principles to predict inheritance patterns and carrier probabilities. In modern research, Mendel's principles are foundational to genome-wide association studies (GWAS) and quantitative trait loci (QTL) mapping, which investigate the genetic basis of traits in populations. In the context of CSIRNet life sciences, questions on Mendelism often test understanding of classical ratios, modifications of these ratios under various genetic interactions, pedigree analysis, linkage and recombination problems, and genotypic probability calculations. High-level understanding includes solving complex cross-outcomes, deducing unknown genotypes from observed phenotypes, and integrating knowledge of epistasis and gene interaction networks. CSIRNet previous year questions on Mendelism. CSIRNet June 2015. In a cross between two heterozygous tall plants TT times TT, what is the expected genotypic ratio of the offspring? Answer: 1 TT, 2 TT, 1 TT. CSIRNet December 2016. Two genes and B interact in such a way that the dominant allele of both is required for a particular phenotype. If both genes are segregating independently, what is the expected phenotypic ratio in the F2 generation? Answer: this is an example of complementary gene interaction 9:7 ratio. CSIRNet June 2014. In a cross between two pea plants with genotype YYRR, heterozygous for seed color and shape, what is the probability of getting offspring with yellow and round seeds dominant traits? Answer: yellow and round equals any genotype with Y underscore and R underscore. Probability equals 3/4 Y underscore times 3/4 R underscore equals 9/16. Let me know the next topic. You can continue with genetic linkage, sex-linked inheritance, or move to molecular biology, biochemistry, or cell cycle. I'll follow the same format in depth. You said. Spontaneity of mutations. ChatGPT said. Here is your CSIRNet JRF Life Sciences Ultra Detailed Lecture on Spontaneity of Mutations, crafted with maximum word limit, no tables, pure paragraph format, and rooted in exam-relevant points, previous year question patterns, and deep theoretical clarity. This is designed as your only study material, thorough, advanced, and tailored to crack the CSIRNet exam. Spontaneity of mutations, concepts, evidence, and molecular basis in evolution and genetics. The spontaneity of mutations refers to the natural and inherent occurrence of genetic changes in the absence of any artificial or environmental induction. This concept stands in contrast to earlier beliefs, particularly those rooted in Lamarckian evolution, which suggested that environmental stress could directly induce adaptive mutations. The concept of spontaneous mutation was a landmark in genetics and molecular biology and is a cornerstone in understanding how genetic variation arises independently of selection pressures. Spontaneous mutations occur randomly during processes such as DNA replication, recombination, or repair, and are not directed by the environment or organismal need. The strongest evidence for the randomness and spontaneity of mutations comes from the famous Luria-Delbruck experiment, 1943, which demonstrated that bacterial resistance to bacteriophages is due to random mutations that occur before exposure, rather than being induced by the virus. They used Escherichia coli and T1 bacteriophage and compared the number of resistant colonies formed when bacteria were exposed to phages either all at once or after independent growth. The fluctuation in mutation frequency across cultures, high variance, indicated that mutations occurred randomly and not in response to phage exposure. This foundational experiment introduced the fluctuation test as a method to detect spontaneous mutations and laid the groundwork for understanding mutagenesis as a stochastic process. On the molecular level, spontaneous mutations can arise due to a variety of reasons, including errors in DNA replication, tautomeric shifts, spontaneous defurination or deamination, and oxidative damage. DNA polymerase, although highly accurate due to its proofreading activity, still makes occasional errors, about 1 in 10 to the power of 9 nucleotides, most of which are corrected by mismatch repair systems. However, some errors escape repair, leading to point mutations, such as transitions and transversions. Tautomeric shifts cause abnormal base pairing, such as a thymine in its rare enol form pairing with guanine instead of adenine, leading to incorrect base incorporation during replication. Similarly, spontaneous deamination of cytosine produces uracil, which if unrepaired, results in C, G to T, A transitions. Depurination, the loss of purine bases A or G creates a basic sites that can lead to misincorporation during replication. Spontaneous mutations also include insertions or deletions indels due to replication slippage, especially in repetitive sequences, leading to frame shifts and functional gene disruptions. Mobile genetic elements, such as transposons, can move within the genome and cause insertions at random loci, often disrupting gene function. All these mechanisms underscore that mutation is a chance event and is not biased toward an organism's future benefit. The spontaneous nature of mutations has deep evolutionary implications. It provides the raw material for natural selection to act upon, aligning perfectly with Darwinian theory, where variation arises independent of the selective environment. Mutations that happen to be advantageous in a given environment may increase in frequency due to selection, but the mutation itself was not caused by the environment. This is a key conceptual distinction that emphasizes the non-Lamarckian basis of evolutionary genetics. Moreover, the rate of spontaneous mutation is under genetic control and varies among organisms, genes, and even within different regions of the genome. For instance, microsatellites have higher mutation rates due to replication slippage, while coding sequences are generally more conserved. Further modern support for the randomness of mutations comes from genomic sequencing and mutation accumulation experiments, which have shown that most mutations are neutral or deleterious and occur without regard to adaptive value. Although recent studies in microbial evolution suggest that certain stress responses can increase mutation rates, known as adaptive mutagenesis or stress-induced mutagenesis, the consensus remains that the mutations themselves are random, even if mutation rates can be modulated under stress. In CSIRNet Life Sciences, questions on this topic typically test understanding of the Luria-Delbruck experiment, types of spontaneous mutations, their molecular mechanisms, the difference between induced and spontaneous mutations, and how mutation feeds into evolutionary theory. A deep grasp of how mutations arise independently of selective pressures is crucial for solving advanced problems in evolution, genetics, and molecular biology. CSIRNet previous year questions on spontaneity of mutations. CSIRNet December 2016. Which of the following experiments provided evidence for the random nature of mutations prior to selection? A Hershey-Chase experiment. Beat Luria-Delbruck fluctuation test. C. Mendelssohn stall experiment. D. Beetle and Tatum's 1 gene 1 enzyme answer. Beat Luria-Delbruck fluctuation test. CSIRNet June 2018. A mutation in a gene occurred without any known exposure to a mutagen. This mutation is best classified as a induced mutation. Beat dominant mutation. C. Spontaneous mutation. D. Artificial mutation answer. C. Spontaneous mutation. CSIRNet December 2020. Which of the following mechanisms is not responsible for spontaneous mutations? A tautomeric shift. B. Ionizing radiation. C. Replication slippage. D. Depurination. Answer. B. Ionizing radiation. This is an induced mutagenic factor. Let me know the next topic. We can go into mutation types, mutagenesis and repair, Hardy-Weinberg equilibrium, or any advanced genetic or molecular biology topic. I will maintain the same level of depth and precision throughout. You said. The evolutionary synthesis. ChatGPT said. Here is your ultra-detailed CSIR Net JRF Life Sciences lecture on the evolutionary synthesis, tailored to the highest standard, deeply grounded in previous year question patterns, and structured as paragraph only, no tables, maximum content density, and built as your only and final coaching material to crack the exam. Every section integrates conceptual, analytical, and application level depth, from base to advanced. The evolutionary synthesis, unifying Darwinian evolution with Mendelian genetics. The evolutionary synthesis, also called the modern synthesis or synthetic theory of evolution, marks one of the most pivotal conceptual breakthroughs in biological science. It was developed primarily between the 1930s and 1950s, when major gaps between Darwin's theory of natural selection and Mendelian genetics were reconciled. Before this synthesis, biologists were divided between Darwinists, who emphasized gradual change in natural selection without a clear understanding of heredity, and Mendelians, who focused on discrete units of inheritance but often dismissed continuous variation in natural selection. The evolutionary synthesis effectively merged Darwin's mechanism of evolution, natural selection, with Mendel's principles of heredity, integrating them under the umbrella of population genetics, thereby forming the foundation of modern evolutionary biology. One of the most significant achievements of the evolutionary synthesis was its establishment of a mathematical and theoretical framework for understanding evolution in terms of changes in allele frequencies in populations over time. This framework was developed through the pioneering works of scientists such as Ronald Fisher, J.B.S. Haldane, and Sewell Wright, who used mathematical models of population genetics to demonstrate how natural selection, mutation, recombination, genetic drift, and gene flow interact to drive evolution. Fisher's 1930 book, The Genetical Theory of Natural Selection, showed how small genetic changes governed by Mendelian inheritance can produce continuous variation seen in populations, thus directly linking microevolutionary processes to Darwinian selection. A critical idea of the synthesis is that evolution is a gradual process resulting from the accumulation of small genetic changes over time, contrary to the earlier saltationist view that new species arise from large, sudden mutations. The continuity between microevolution changes within populations and macroevolution formation of new species and higher taxa was established, highlighting that speciation could result from the gradual divergence of populations under the influence of evolutionary forces. Additionally, the synthesis clarified the central role of populations as the units of evolution, with natural selection acting on individuals but evolution occurring at the population level through changes in gene frequencies. Another core component was the refinement of the species concept, especially the biological species concept proposed by Ernst Mayr, which defines species as groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. This concept emphasized the role of reproductive isolation and gene flow in the maintenance and origin of species. Similarly, Theodosius Dobzhansky's work in genetics and natural populations, especially his 1937 book Genetics and the Origin of Species, was instrumental in demonstrating that mutation and recombination provide raw material for selection, and that genetic diversity exists in natural populations, even in the absence of selection. The evolutionary synthesis also incorporated insights from paleontology, systematics, and comparative morphology, thanks to contributions from scientists like George Gaylord Simpson and G.G. Simpson, who integrated fossil records into evolutionary theory. They showed that patterns in the fossil record, such as gradualism and adaptive radiation, could be explained by synthetic evolutionary mechanisms. Simpson's work proved that macroevolutionary patterns could be interpreted as the cumulative result of microevolutionary changes extended over geological time. One of the strengths of the synthesis was its ability to reject both Lamarckism, inheritance of acquired traits, and mutationism, that mutations alone drive evolution, providing instead a multifactorial explanation where mutation introduces variation, recombination shuffles alleles, and natural selection acts on phenotypes to shape genetic composition. Moreover, genetic drift, though initially underemphasized, was later recognized, especially by Wright, as playing a substantial role in small populations. Despite its strengths, the evolutionary synthesis had limitations. It initially underappreciated areas like developmental biology, epigenetics, and phenotypic plasticity, which are now important in the extended evolutionary synthesis. Nonetheless, its original framework remains foundational and is essential to understanding CSIR net level questions in evolution and population biology. CSIR net previous year questions on the evolutionary synthesis. CSIR net December 2015. Which of the following concepts was fundamental to the modern synthesis of evolution? Inheritance of acquired characters. B. Gene flow and genetic drift are not significant in evolution. C. Evolution occurs primarily through large mutations. D. Natural selection operates on heritable variation. Answer. D. Natural selection operates on heritable variation. CSIR net June 2019. The modern synthesis unified which two major biological ideas? Lamarckism and paleontology. B. Mutation theory and embryology. C. Darwinian evolution and Mendelian genetics. D. Intelligent design and molecular biology. Answer. C. Darwinian evolution and Mendelian genetics. CSIR net December 2021. The concept that evolution proceeds through the change in allele frequency within a population over time is best described by Mendelian inheritance. B. Hardy-Weinberg equilibrium. C. Synthetic theory of evolution. D. Intelligent evolution. Answer. C. Synthetic theory of evolution. Let me know the next topic. I can continue with Hardy-Weinberg principle, genetic drift versus natural selection, speciation, or go deep into molecular evolution or developmental constraints in evolution. This is your full-fledged coaching. I'll handle everything with this same level of detail. You said. Origin of basic biological molecules. ChatGPT said. Here is your ultra-detailed CSIR net JRF Life Sciences lecture on the topic, Origin of basic biological molecules, written in maximum word limit, paragraph form only, and tailored to the CSIR net exam with JRF level precision. It includes deep conceptual and applied knowledge, historical and molecular context, and previous year questions with answers in years mentioned, crafted to be your only study material, no other reference needed. Origin of basic biological molecules, chemical evolution before life. The origin of basic biological molecules is a central theme in the field of biogenesis, which explores how non-living chemical substances gave rise to the first biomolecules that form the foundation of life on Earth. This topic deals with the transition from simple inorganic compounds to complex, carbon-based macromolecules such as amino acids, nucleotides, sugars, and lipids. The widely accepted scientific framework for this process is the chemical evolution theory, which states that life originated through a gradual chemical process in a primitive earth environment that was conducive to the formation of organic molecules under abiotic conditions. The earliest and most influential hypothesis in this domain was proposed by Alexander Perrin, 1924, and J.B.S. Haldane, 1929, who independently suggested that life began in a primordial soup of organic molecules formed under reducing atmospheric conditions. They posited that Earth's early atmosphere lacked free oxygen but was rich in gases like methane, CH, ammonia, NH, hydrogen, H, and water vapor, HO, which facilitated the synthesis of organic compounds when exposed to energy sources such as lightning, ultraviolet radiation, or volcanic activity. These energy sources drove abiotic chemical reactions among the simple gases, producing small organic molecules that accumulated in the oceans. The experimental validation of this theory came in 1953 through the landmark Miller-Urey experiment, in which Stanley Miller and Harold Urey simulated early Earth conditions using a closed system containing water, methane, ammonia, and hydrogen. When exposed to electrical sparks simulating lightning, the system produced a variety of amino acids, such as glycine, alanine, and aspartic acid, within days. This experiment provided the first empirical evidence that biological molecules can arise spontaneously under prebiotic conditions, lending strong support to chemical evolution as the basis of life's origin. Following amino acids, the origin of nucleotides, the building blocks of RNA and DNA, has also been a subject of intensive study. Prebiotic synthesis of purines and pyridines under early Earth-like conditions has been demonstrated, although the spontaneous assembly of complete nucleotides is chemically more complex than amino acid synthesis. Nonetheless, John Sutherland and colleagues have shown plausible pathways for the abiotic synthesis of activated nucleotides under prebiotic conditions, involving intermediates like cyanamide, glycolaldehyde, and phosphate. Sugars, such as ribose, may have formed through the Formose reaction, where formaldehyde HCHO polymerizes under alkaline conditions in the presence of basic catalysts like calcium hydroxide. However, ribose is chemically unstable, especially under high temperature and pH fluctuations, which challenges its accumulation in prebiotic conditions. This has led to alternative theories such as RNA world hypotheses where simpler or more stable sugar backbones may have preceded ribose. The formation of lipids and protocellular membranes is also a critical step in the emergence of life. Fatty acids can spontaneously form micelles and liposomes in aqueous environments, thereby creating structures capable of compartmentalizing biochemical reactions. Fatty acids have been found in meteorites and can form biotically through Fischer-Tropsch type synthesis, which requires simple gases like CO and H on metal catalysts under high pressure and temperature. Collectively, the spontaneous origin of basic biological molecules is now understood as a multi-step process of chemical evolution, where small molecules gradually combine to form increasingly complex and self-organizing structures. Several alternative theories have been proposed, such as the iron-sulfur world hypothesis, where metabolic precursors formed on the surface of metal sulfides near hydrothermal vents, and the panspermia hypothesis, which posits that organic molecules or microbial life may have been delivered to Earth via meteorites. Despite such alternatives, the consensus remains that life's molecular building blocks can form biotically under favorable conditions. In modern evolutionary biology, the origin of basic molecules is not just historical but also fundamental to understanding the emergence of metabolism, pre-RNA world systems, and the origin of information-carrying molecules. The topic links directly to research in astrobiology, systems chemistry, and origin of life studies. In the CSIR-NET exam, this topic is often tested in the context of chemical evolution, Miller-Urey experiments, composition of early atmosphere, and origin of biological macromolecules. CSIR-NET previous year questions on origin of basic biological molecules. CSIR-NET December 2016. Which of the following was a key outcome of the Miller-Urey experiment? A demonstration of gene expression in bacteria. B. Isolation of mitochondria from eukaryotic cell. C. Synthesis of amino acids under simulated prebiotic conditions. D. Discovery of the double helix structure of DNA. Answer. C. Synthesis of amino acids under simulated prebiotic conditions. CSIR-NET June 2020. Which of the following gases were included in the original Miller-Urey experiment to simulate early Earth's atmosphere? A. O. CO. H. O. N. B. CH. NH. H. HOC. CO. HS. O. NHD. N. O. CH. CO. Answer. B. CH. NH. H. HO. CSIR-NET December 2018. Which of the following hypotheses is supported by the prebiotic formation of cell-like vesicles in laboratory simulations? A. Iron-sulfur world hypothesis. B. Panspermia hypothesis. C. Endosymbiotic theory. D. Lipid world hypothesis. Answer. D. Lipid world hypothesis. Let me know if you'd like to move on to RNA world hypothesis, coacervates and protobionts, or molecular evolution next. I will continue this format: rich, analytical, paragraph-based, exam-oriented, and your final and complete preparation source. You said. Abiotic synthesis of organic monomers. ChatGPT said. Here is your ultra-detailed CSIR-NET JRF level theory lecture on abiotic synthesis of organic monomers, written in maximum word limit, paragraph-based, with no tables, and tailored with previous year question patterns in mind. This lecture connects the chemical evolution of life to the origin of amino acids, nucleotides, sugars, and lipids under prebiotic conditions, which is a frequently tested concept in CSIR-NET life sciences. Abiotic synthesis of organic monomers. The abiotic synthesis of organic monomers refers to the formation of biologically relevant molecules, such as amino acids, nucleotides, sugars, and fatty acids, through non-biological chemical processes in pre-life Earth conditions. This phenomenon lies at the heart of the chemical origin of life and constitutes the first step in abiogenesis, where life's fundamental building blocks were formed under natural, energy-driven reactions, independent of enzymatic or cellular machinery. The process involves simple inorganic compounds like methane, CH, ammonia, NH, hydrogen, H, water vapor, HO, carbon dioxide, CO, and nitrogen, N, which, when subjected to external energy sources such as lightning, ultraviolet UV radiation, volcanic activity, or hydrothermal vent heat, give rise to progressively complex organic compounds. The experimental foundation of this concept was laid by the Miller-Urey experiment, 1953, which remains one of the most iconic studies in origin of life research. In this experiment, Stanley Miller and Harold Urey mimicked the early Earth's reducing atmosphere using a closed system containing water, methane, ammonia, and hydrogen. They introduced electric sparks to simulate lightning and cycled the mixture through water and a condenser. After several days, they observed the spontaneous formation of various amino acids, such as glycine, alanine, and aspartic acid, along with other organic compounds. This demonstrated for the first time that amino acids, the monomers of proteins, can form under abiotic conditions in an environment that simulates primitive Earth. Following amino acids, researchers have investigated the abiotic synthesis of nucleotides, which are essential for the formation of RNA and DNA. The challenge with nucleotide synthesis lies in the complexity of their structure, which consists of a nitrogenous base, a sugar ribose or deoxyribose, and a phosphate group. Although early attempts failed to produce full nucleotides, later studies, such as those by John Sutherland and team, successfully synthesized activated nucleotides via prebiotic pathways involving cyanamide, glycolaldehyde, and inorganic phosphate, all in aqueous environments. Their work provided strong support for the RNA world hypothesis, in which RNA or RNA-like molecules emerge as early information carriers and catalytic entities. The abiotic synthesis of sugars is explained via the Formose reaction, first described by Alexander Butlerov in 1861. This reaction involves the polymerization of formaldehyde HCHO into sugars like ribose and other pentoses and hexoses in the presence of alkaline conditions and divalent metal ions such as Ca². However, ribose is chemically unstable under prebiotic conditions, and this instability poses a problem for the accumulation of nucleotides. As a result, some hypotheses suggest that other more stable sugars might have preceded ribose in early nucleic acid-like structures. Lipids, particularly fatty acids, are crucial for the formation of primitive cell membranes. Fatty acids can be synthesized abiotically through Fischer-Tropsch type reactions, where carbon monoxide CO and hydrogen react over metal catalysts like iron or nickel at high temperatures and pressures. These reactions can produce long-chain hydrocarbons, including fatty acids, which spontaneously assemble into micelles and liposomes in aqueous solutions. Such structures resemble the first protocells capable of enclosing molecules, thus creating distinct internal chemical environments, a vital step in the development of cellular life. Another major class of organic monomers includes purines and pyrimidines, the nitrogenous bases of nucleic acids. The synthesis of purines like adenine has been demonstrated in laboratory conditions by heating hydrogen cyanide (HCN) in aqueous solutions. Adenine, for instance, can be synthesized by the condensation of 5 molecules of HCN, a process that may have occurred in pools of evaporating water exposed to sunlight. The formation of pyrimidines is more complex but has also been demonstrated under simulated prebiotic conditions. The sources of energy that facilitated these synthetic reactions include not just lightning, as in the Miller-Urey setup, but also UV radiation from the sun, radioactivity from Earth's crust, heat from volcanic vents and hydrothermal systems, and even shock energy from asteroid impacts. These diverse sources would have created a range of chemical environments across the Earth's surface and oceans, each potentially contributing to the formation of different types of organic monomers. The abiotic synthesis of organic monomers is also supported by evidence from extraterrestrial sources, such as carbonaceous chondrites, meteorites like the Murchison meteorite, which contain amino acids, sugars, and even nucleobase-like compounds. This suggests that such monomers may not only form on Earth but could also be ubiquitous in the universe, forming wherever suitable chemical and energy conditions exist. In summary, the abiotic synthesis of organic monomers involves a multi-step and diverse set of chemical processes that produce the first biomolecular components of life from simple inorganic precursors. These monomers provide the raw materials for the eventual formation of polymers, proteins, nucleic acids, etc., and more complex biological systems. The field is closely linked to prebiotic chemistry, planetary science, and molecular evolution. For CSIR-NET Life Sciences, this topic is foundational and appears in questions regarding chemical evolution, prebiotic synthesis, experimental validation, Miller-Urey, and the molecular building blocks of early life. CSIR-NET previous year questions on abiotic synthesis of monomers. CSIR-NET December 2017. The Miller-Urey experiment provided the first evidence that ribosomes could form spontaneously. RNA could replicate without enzyme. C. Organic molecules such as amino acids can be synthesized abiotically. D. Life began near deep sea hydrothermal vents. Answer. C. Organic molecules such as amino acids can be synthesized abiotically. CSIR-NET June 2019. Which of the following is a key product of the Formose reaction relevant to prebiotic chemistry? A. Adenine. B. Ribose. C. Glycine. D. Urea. Answer. B. Ribose. CSIR-NET December 2020. Which condition is not essential for the Miller-Urey type abiotic synthesis of organic molecules? A. Presence of methane and ammonia. B. Presence of a reducing atmosphere. C. Continuous UV radiation. D. An energy source like lightning. Answer. C. Continuous UV radiation. Let me know if you'd like to continue with polymerization of monomers to macromolecules, formation of protocells coacervates, micelles, or RNA world hypothesis. Next. I'll maintain the same high standard and exam-oriented depth for each topic. You said, same of polymers, ChatGPT said. Here is your CSIR Net JRF level, ultra-detailed theory lecture on polymerization of monomers to macromolecules, written in maximum word limit, without tables, and focused on previous year question trends. This topic links directly to the origin of life, covering how basic monomers form complex macromolecules like proteins, nucleic acids, and polysaccharides under prebiotic conditions. Polymerization of monomers to macromolecules. Polymerization of monomers into macromolecules marks a critical transition in the origin of life, bridging the gap between the chemical and biological stages of evolution. While the abiotic synthesis of organic monomers such as amino acids, nucleotides, and simple sugars provided the building blocks, it is the non-enzymatic polymerization of these into functional polymers like proteins, nucleic acids, and polysaccharides that allow the emergence of biological complexity and self-replicating systems. These macromolecules lay the foundation for catalysis, genetic information storage, and structural organization, the hallmarks of living systems. The challenge in prebiotic chemistry is that polymerization in aqueous environments is thermodynamically unfavorable due to the requirement of water elimination condensation reactions. Therefore, various experimental models have been proposed to overcome this barrier, simulating environmental conditions that could have facilitated dehydration synthesis on the primitive Earth. One prominent hypothesis involves drying heating cycles at geothermal sites or tidal pools. For example, when amino acids are exposed to heat on mineral surfaces like montmorillonite clay or basaltic rock, peptide bonds can form through thermal condensation, leading to the synthesis of short peptides, oligopeptides, without the aid of enzymes. Mineral surfaces play a pivotal role in catalyzing prebiotic polymerization. Montmorillonite clay, in particular, has been shown to promote the polymerization of nucleotides into RNA-like polymers. This is because the clay surface can absorb nucleotides, orient them in close proximity, and lower the activation energy required for phosphodiester bond formation. Furthermore, the layered structure of clay minerals may have offered protection against hydrolysis and UV radiation, thereby stabilizing the newly formed polymers. Another critical aspect of non-enzymatic polymerization is the use of activated monomers. In modern biochemistry, polymerization is driven by high-energy molecules like ATP and GTP. In prebiotic chemistry, analogs of these molecules were likely formed, such as phosphoramides or other prebiotically plausible nucleotide triphosphates, which act as energy-rich intermediates capable of facilitating RNA strand elongation. Studies by Leslie Orgel and others have shown that activated ribonucleotides can polymerize on clay surfaces or in eutectic phases to form RNA chains of considerable length and complexity. Peptide bond formation has also been demonstrated under plausible prebiotic conditions. Experiments have shown that amino acids, when subjected to cycles of drying and heating, especially in the presence of carbonyl sulfide, a volcanic gas, can spontaneously condense to form short peptides. Other facilitating environments include hydrothermal vents, where fluctuating temperatures and mineral-rich surroundings create conditions conducive for polymer formation. These peptides might have served as primitive enzymes, protoenzymes, catalyzing further chemical reactions essential for early metabolism. The polymerization of sugars into polysaccharides under abiotic conditions is more complex and less understood. However, it is believed that certain metal ions in drying heating cycles could promote the glycosidic bond formation between monosaccharides, producing oligosaccharides. Though less explored experimentally, the role of carbohydrates in stabilizing nucleic acid backbones or forming early structural components cannot be dismissed. An important phenomenon associated with these prebiotic polymers is autocatalysis, where a polymer promotes its own formation or assists in the formation of similar molecules. This concept is central to the RNA world hypothesis, which proposes that RNA molecules were the first self-replicating entities, capable of both storing genetic information and catalyzing reactions. The discovery of ribozymes, RNA molecules with catalytic activity, has added substantial support to this theory. It suggests that once polymerization led to a sufficiently long RNA strand with catalytic capability, it could direct its own replication, leading to Darwinian evolution at the molecular level. Further, protocell formation, the enclosure of these macromolecules within lipid vesicles or coacervates, would have provided physical compartmentalization, concentrating reactants and protecting genetic material. Within these protocells, polymerization reactions might have become more efficient, eventually leading to the emergence of primitive metabolic pathways and selection processes. In conclusion, the polymerization of monomers into macromolecules under prebiotic conditions represents a pivotal event in the emergence of life. These reactions, although inefficient by modern standards, could have occurred in a variety of natural environments such as geothermal fields, hydrothermal vents, tidal flats, or even icy comets. The prebiotic formation of proteins, RNA-like molecules, and polysaccharides established the functional and informational foundation upon which natural selection could act. In CSIR Net Life Sciences, this topic is essential to understanding the origin of biological systems from chemical evolution, and questions often test conceptual understanding of experimental models, e.g., Orgel, ferrous energy requirements, and catalytic surfaces. CSIR Net previous year questions on polymerization of macromolecules. CSIR Net June 2016. Which of the following mineral surfaces is known to facilitate abiotic polymerization of RNA under prebiotic conditions? A. Silicon dioxide. B. Graphite. C. Montmorillonite clay. D. Hematite. Answer: C. Montmorillonite clay. CSIR Net December 2018. The polymerization of amino acids into peptides under prebiotic conditions is favored by a dilute aqueous solution. B. Presence of high energy phosphate. B. Presence of high energy phosphate bond. C. Wet dry cycles and mineral surfaces. D. Enzymes. Answer: C. Wet dry cycles and mineral surfaces. CSIR Net June 2021. Activated nucleotides are essential for RNA polymerization in prebiotic chemistry because a. They inhibit hydrolysis. B. They lower the melting temperature of RNA. C. They provide energy for forming phosphodiester bonds. D. They bind to lipids for membrane synthesis. Answer: C. They provide energy for forming phosphodiester bonds. Let me know if you'd like to proceed with formation of protocells or RNA world hypothesis next, and I'll continue with the same level of detail in CSIR Net focus. You said polymers. ChatGPT said. Here is your ultra-detailed CSIR Net JRF level lecture on the topic polymers in the context of the origin of life, written in maximum word limit, structured in paragraph format, and specifically tailored for the CSIR Net Life Sciences syllabus. This note includes no tables, follows the from base to advanced approach, and concludes with previous year questions with answers and years mentioned. This is made to be your complete and only study material for cracking the exam. Polymers: origin, structure, and significance in the origin of life. The concept of polymers is central to the understanding of life, both in contemporary biology and in the context of its origin on Earth. A polymer is a large molecule composed of repeating structural units, monobonded together. In biological systems, major polymers include proteins, polymers of amino acids, nucleic acids, polymers of nucleotides, and polysaccharides, polymers of sugars. These macromolecules perform the essential tasks of catalysis, information storage, energy management, and structural integrity, making their prebiotic origin a crucial step in the emergence of life. In the early Earth environment, simple organic molecules were likely formed via abiotic synthesis, e.g., Miller-Urey experiment. However, for life to emerge, these monomers had to form polymers that could retain information, catalyze reactions, and self-replicate. The polymerization of monomers under prebiotic conditions, however, poses a significant biochemical and thermodynamic challenge. Polymer formation typically involves condensation reactions, where water is removed as monomers are joined, yet this is thermodynamically unfavorable in an aqueous environment. To overcome this, several prebiotic models have been proposed, including wet dry cycles on terrestrial volcanic surfaces, tidal flats, or mineral surfaces such as montmorillonite clay and basaltic rock. These surfaces can absorb monomers, orient them in proximity, and facilitate their polymerization. Studies have shown that clay minerals can catalyze the formation of short RNA polymers, indicating their possible role in the origin of informational macromolecules. Similarly, peptides have been shown to form when amino acids are subjected to dry heat cycles, sometimes aided by gases like carbonyl sulfide or activated condensing agents. The most significant polymers in the context of prebiotic evolution are nucleic acids, particularly RNA, which is hypothesized to be the first self-replicating informational polymer. RNA not only stores genetic information but also performs catalytic functions, ribozymes making it a dual-function polymer suited for early life forms. The RNA world hypothesis posits that life began with RNA-based systems that later evolved to DNA and proteins. The ability of RNA polymers to form through template-directed, non-enzymatic polymerization of activated nucleotides further strengthens this theory. For example, phosphorinidazolide activated nucleotides can polymerize in the presence of Mg squared and mineral surfaces to form RNA strands. Proteins, made from amino acid polymers, are another class of essential macromolecules. Even in the absence of genetic templates, random polypeptides could have formed under plausible prebiotic conditions. These may have acted as protoenzymes, rudimentarily catalyzing simple biochemical reactions. This would increase local reaction rates in favor of chemical evolution of more efficient systems. Polysaccharides, although often overlooked in early life models, are crucial for providing structural scaffolds, energy reserves, and potential roles in encapsulating other polymers. The polymerization of monosaccharides to form glycosidic bonds under abiotic conditions is more complex but not impossible. Experiments suggest that metal ions and dehydration synthesis could allow the formation of oligosaccharides, which may have played roles in the formation of early membranes or coacervates, contributing to compartmentalization and protection of catalytic systems. In the broader context, polymerization of monomers into macromolecules under prebiotic earth conditions represents a transition from chemistry to biology. Once such polymers began interacting, replicating, and mutating, Darwinian evolution could take hold, giving rise to increasingly complex biochemical systems. The formation and role of polymers, thus, mark the origin of function, heredity, and biological individuality. Without polymers, there could be no enzymes, no replication, no metabolism, and ultimately, no life. In the CSIR Net Life Sciences examination, this topic is frequently tested, especially in Section C, higher order thinking, with questions targeting understanding of prebiotic chemistry, experimental models of polymer synthesis, the RNA world, and the transition to early protocellular life. Students should be able to integrate biochemical principles with evolutionary theory and prebiotic earth models. CSIR Net previous year questions on polymers and origin of life. CSIR Net December 2014. Which of the following is considered as a possible catalytic surface for polymerization of RNA in the prebiotic earth environment? A quartz. B. Hematite. C. Montmorillonite clay. D. Limestone. Answer. C. Montmorillonite clay. CSIR Net June 2019. The importance of RNA as a polymer in the origin of life hypothesis is due to its role in photosynthesis. B. Ability to store information and catalyze reactions. C. Structural rigidity. D. Strong base pairing with DNA. Answer. B. Ability to store information and catalyze reactions. CSIR Net December 2022. Which property of polymers is most critical for their role in the origin of life? A. High solubility in water. B. Ability to form crystals. C. Capability of self-replication and catalysis. D. Resistance to UV radiation. Answer. C. Capability of self-replication and catalysis. Let me know if you'd like to continue with formation of protocells, RNA world hypothesis, or origin of genetic code, all of which are connected to this section and heavily asked in CSIR Net JRF. You said. Concept of a parent and Haldane. ChatGPT said. Here is your ultra-detailed JRF level lecture on the concept of a parent and Haldane, specifically tailored for CSIR Net Life Sciences preparation. The content is structured in dense paragraph format, contains maximum word limit, avoids tables, and includes 2 to 3 previous year questions with answers and years mentioned. This note is created to serve as your one and only source for mastering this topic for the exam. Concept of a parent and Haldane, foundation of chemical evolution theory. The apparent Haldane hypothesis is a foundational concept in evolutionary biology and the origin of life studies. Formulated independently in the 1920s by Alexander I Apparent, a Russian biochemist, and J.B.S. Haldane, a British geneticist and evolutionary biologist, this theory laid the groundwork for understanding the chemical evolution of life on Earth. At its core, the apparent Haldane model suggests that life originated not through mystical or spontaneous generation but through a gradual, stepwise chemical process in Earth's primitive environment, driven by physical and chemical laws. This concept marks the first scientific attempt to explain abiogenesis, the origin of life from non-living matter, using empirical principles and without invoking supernatural explanations. According to apparent and Haldane, the early Earth, approximately 4.5 billion years ago, had a reducing atmosphere rich in gases such as methane, CH, ammonia, NH, hydrogen, H, and water vapor, HO, but lacking free oxygen, O. This atmosphere was significantly different from the present-day oxidizing environment. The reducing condition was critical, as it allowed the accumulation of complex organic molecules without rapid degradation by oxidation. Both scientists propose that energy sources such as UV radiation, lightning, and geothermal heat catalyze chemical reactions between the atmospheric gases, leading to the synthesis of simple organic compounds, such as amino acids, sugars, nucleotides, and fatty acids. These organic monomers likely accumulated over time in Earth's primitive oceans, forming a primordial soup, a rich aqueous mixture of organic compounds. This term was first used by Haldane, who emphasized the oceans as a site for chemical evolution. In this soup, organic molecules would undergo further reactions, eventually forming polymers like proteins and nucleic acids. Apparent went further to propose that these polymers could aggregate into coacervates, microscopic droplets surrounded by boundary-like interface that could concentrate and organize biochemical reactions. These coacervates are protocells represented for cellular structures that exhibited some life-like properties such as growth, division, and selective uptake of molecules from the environment. A key assumption of their hypothesis was the absence of enzymes or biological catalysts in the early stages. Therefore, all initial reactions were non-enzymatic and purely chemical, driven by random interactions and aided by environmental conditions. This idea anticipates the RNA world hypothesis, which emerged much later, suggesting that the first self-replicating systems may have involved RNA molecules with catalytic activity. However, apparent and Haldane did not specify a genetic or information-storing molecule. Instead, they emphasized the gradual complexity increase from chemical to biological systems. Another critical aspect of their theory is the concept of chemical evolution preceding biological evolution. The process was envisioned to occur over millions of years, allowing the accumulation of diversity and complexity. Eventually, systems emerged that could replicate, mutate, and undergo natural selection, thereby initiating biological evolution. In this way, the apparent Haldane hypothesis bridges chemistry and Darwinian evolution, making it a precursor to the modern synthesis. The significance of this theory was later supported experimentally by the Miller-Urey experiment, 1953, which simulated early Earth conditions and demonstrated the spontaneous synthesis of amino acids from simple gases using electric sparks. Although modern understanding has refined many details, such as the composition of the primitive atmosphere and the role of hydrothermal vents, the apparent Haldane hypothesis remains a pivotal theoretical framework in studies of the origin of life. From the perspective of CSIR Net Life Sciences, this topic is critical because it connects molecular biology, biochemistry, and evolutionary biology, all of which are major components of the syllabus. Questions often focus on the nature of the early atmosphere, mechanisms of abiotic synthesis, role of energy sources, and experimental validation such as the Miller-Urey experiment and formation of coacervates. You must understand this theory not only historically but also in relation to subsequent models like the RNA world and the evolution of protocells. Previous year CSIR Net questions. CSIR Net June 2012. According to the apparent Haldane hypothesis, life on Earth arose from a spontaneous generation in modern conditions via pre-existing organisms. C. Gradual chemical evolution in a reducing atmosphere. D. Extraterrestrial panspermia. Answer. C. Gradual chemical evolution in a reducing atmosphere. CSIR Net December 2015. Which of the following energy sources was proposed by apparent Haldane to have facilitated the abiotic synthesis of organic molecules on primitive Earth? A. Nuclear fission. B. UV radiation and lightning. C. Photosynthesis. D. Radio waves. Answer. B. UV radiation and lightning. CSIR Net June 2018. The concept of primordial soup, where organic molecules accumulated and evolved into complex systems, was originally proposed by A. Stanley Miller. B. Charles Darwin. C. J.B.S. Haldane. D. Francis Crick. Answer. C. J.B.S. Haldane. Would you like to continue with the next topic, Miller-Urey experiment, formation of coacervates, or RNA world hypothesis? These are directly linked and frequently asked in CSIR Net JRF. You said. Experiment of Miller 1953 ChatGPT said. Here is your CSIRNet JRF level ultra detailed lecture on the Miller 1953 experiment, created with maximum word limit, dense paragraph structure, no tables, and tailored to match the pattern and depth of previous CSIRNet questions. This lecture is designed to be your only study material for mastering this topic and cracking the exam. Miller 1953 experiment, demonstration of abiotic synthesis of organic molecules. The Miller 1953 experiment, also known as the Miller-Urey experiment, is one of the most iconic and foundational studies in the field of origin of life research. Conducted by Stanley L. Miller, a graduate student under the supervision of Harold C. Urey at the University of Chicago, this experiment was the first to provide empirical evidence supporting the apparent Haldane hypothesis of chemical evolution. The goal of the experiment was to simulate the prebiotic conditions of early Earth and test whether organic compounds essential to life could have formed spontaneously from inorganic precursors in a reducing atmosphere. Miller constructed a closed glass apparatus that mimicked the hypothesized environment of primitive Earth. The setup included a boiling flask containing water representing the ancient oceans, which was connected to a second flask filled with a mixture of gases: methane, CH, ammonia, NH, hydrogen, H, and water vapor, HO. This combination simulated the reducing atmosphere assumed to have existed on Earth more than 3.5 billion years ago. A pair of electrodes was installed in the gas chamber to generate continuous electrical sparks, simulating lightning, one of the proposed natural energy sources on the primitive Earth. The vapor was continuously circulated through the system, condensed by a cooling coil, and collected in a trap designed to accumulate newly formed compounds. After running the experiment for 1 week, Miller analyzed the contents of the collection trap and discovered a variety of organic compounds, including several amino acids such as glycine, alanine, aspartic acid, and glutamic acid. Amino acids are the building blocks of proteins, and their abiotic synthesis in the laboratory was a groundbreaking confirmation that organic molecules can be formed under prebiotic conditions without any biological intervention. Miller also detected other simple organic molecules, including hydrocarbons and urea, indicating that complex chemical evolution was indeed possible under the right environmental conditions. The significance of this experiment was manifold. First, it provided strong support for the chemical evolution theory, suggesting that the origin of life could have been preceded by a long period of prebiotic organic synthesis. Second, it showed that simple molecules present in early Earth's atmosphere, when subjected to the right energy inputs, could self-organize into biologically relevant compounds. Third, it offered a testable, replicable model for studying the origin of biomolecules, opening the door for numerous follow-up experiments that have expanded our understanding of prebiotic chemistry. Finally, it demonstrated the power of laboratory simulations to answer questions that were previously thought to be purely speculative or philosophical. However, subsequent research has led to some revisions of Miller's assumptions. Later geological evidence suggested that the primitive Earth's atmosphere may not have been as strongly reducing as Miller proposed. Instead, it might have been weakly reducing or even neutral, composed largely of carbon dioxide, CO, nitrogen, N, and water vapor. Yet, later experiments have shown that even under less reducing conditions or in the presence of volcanic gases or hydrothermal vents, similar organic molecules can still be synthesized. Additionally, researchers have discovered that meteoritic material, such as that found in the Murchison meteorite, also contains a wide range of amino acids, supporting the idea that organic synthesis could occur in extraterrestrial environments as well. The Miller experiment also inspired later models such as the RNA world hypothesis, the concept of autocatalytic cycles, and theories involving mineral surface catalysis, but it remains a foundational experiment that every serious life science student must understand thoroughly. In the context of CSIRNet JRF Life Sciences, this experiment often appears in questions testing your understanding of prebiotic chemistry, experimental biology, and evolution of biomolecules. Questions may focus on the design of the experiment, the gases used, the types of molecules formed, the energy source, and the interpretations and limitations of the findings. Previous year CSIRNet questions. CSIRNet June 2011. Which one of the following gases was not used in the Miller's experiment simulating primitive Earth conditions? A. Methane. B. Hydrogen. C. Oxygen. D. Ammonia. Answer. C. Oxygen. CSIRNet December 2014. Previous year CSIRNet questions. CSIRNet June 2011. Which one of the following gases was not used in the Miller's experiment simulating primitive Earth conditions? A. Methane. B. Hydrogen. C. Oxygen. D. Ammonia. Answer. C. Oxygen. CSIRNet December 2014. The major significance of Miller's experiment was that it A. Disproved the theory of evolution. B. Demonstrated abiotic synthesis of amino acids. C. Proved the existence of life on Mars. D. Provided evidence for panspermia. Answer. B. Demonstrated abiotic synthesis of amino acids. CSIRNet June 2017. The source of energy used in Miller's experiment to mimic early Earth's atmosphere was A. Heat from magma. B. Sunlight. C. Electrical discharge. D. UV rays. Answer. C. Electrical discharge. Would you like to continue with the next high-yield topic, formation of coacervates and protocells, RNA world hypothesis, or evolution of prokaryotes and eukaryotes? These are also heavily asked areas in CSIRNet JRF. You said, the first cell, ChatGPT said. Here is your CSIRNet JRF level, ultra detailed, maximum coverage lecture on the first cell, crafted precisely to match CSIRNet Life Sciences exam standards. This lecture is presented in dense paragraph form, without tables, and includes 2 to 3 previous year questions with answers in your mentioned. It is written to be your exclusive, complete study material, from base to advanced level, for cracking this topic in the exam. The first cell, the origin of life's first biological entity. The emergence of the first cell marks one of the most profound transitions in the history of life on Earth, the moment when non-living chemistry evolved into living biology. This transformation is the final and most crucial phase of the chemical evolution hypothesis, where complex organic molecules self-organize into a system capable of metabolism, growth, reproduction, and evolution. The first cell is believed to have arisen approximately 3.8 to 4 billion years ago, shortly after the formation of the Earth and the stabilization of its oceans. Understanding the origin of the first cell requires integrating insights from molecular biology, biochemistry, geology, and evolutionary theory. Before the appearance of the first true cell, the Earth likely harbored a variety of precellular structures such as coacervates, microspheres, and lipid vesicles. These structures could form spontaneously from amphiphilic molecules, e.g., fatty acids, phospholipids, in aqueous environments. They exhibit some life-like properties such as compartmentalization and selective permeability, making them prime candidates for protocells, transitional entities between non-life and life. Such protocells may have concentrated biochemical reactions, enhanced molecular interactions, and protected catalytic molecules like RNA from degradation. A major breakthrough in our understanding of the first cell came with the development of the RNA world hypothesis, which proposes that RNA was the first genetic and catalytic molecule. RNA possesses the unique ability to store genetic information and catalyze chemical reactions, ribozymes. It is hypothesized that within these protocells, RNA molecules capable of self-replication and basic catalysis emerge through a series of non-enzymatic chemical reactions. This led to natural selection at the molecular level, favoring RNA sequences that could replicate more efficiently or perform useful reactions, such as amino acid activation or peptide bond formation. The transition from RNA-based systems to more stable and complex DNA-protein systems was likely gradual. DNA eventually replaced RNA as the primary information storage molecule due to its chemical stability, while proteins, with diverse catalytic and structural capabilities, became the dominant catalysts. The first cell thus evolved from a primitive protocell containing self-replicating RNA to a more organized entity with a lipid bilayer membrane, DNA genome, ribosomes, and enzymatic pathways for metabolism. The metabolism of the first cells was likely anaerobic and heterotrophic, relying on organic molecules available in the primordial environment, a concept called the heterotroph hypothesis. Only later did autotrophic pathways such as chemosynthesis and photosynthesis evolve, enabling organisms to generate their own organic matter and drastically altering Earth's atmosphere. The emergence of photosynthetic prokaryotes, e.g., cyanobacteria, released oxygen as a byproduct, leading to the great oxygenation event and paving the way for aerobic respiration and eukaryotic evolution. Structurally, the first true cells were likely prokaryotic, lacking a nucleus and membrane-bound organelles. Fossil evidence, such as stromatolites dated to over 3.5 billion years ago, supports the early presence of microbial life. These cells were simple but remarkably efficient, possessing plasma membranes, genetic material, ribosomes, and rudimentary metabolic pathways. Through mutation, replication, and selection, these primitive organisms diversified and evolved into the vast microbial world that dominates Earth even today. From a CSIRNet Life Sciences standpoint, understanding the origin of the first cell is vital because it connects key concepts across molecular evolution, genetics, origin of life, and cell biology. Commonly asked questions involve the composition and properties of protocells, the role of RNA, the transition from chemical to biological evolution, and the features of the earliest life forms. Previous year CSIR Net questions. CSIR Net December 2012. Which one of the following best describes the earliest living cells on Earth? A. Aerobic and photosynthetic. B. Anaerobic and heterotrophic. C. Aerobic and autotrophic. D. Eukaryotic and heterotrophic. Answer. B. Anaerobic and heterotrophic. CSIR Net June 2016. The RNA world hypothesis proposes that a proteins evolved before nucleic acids. B. DNA was the first genetic material. C. RNA served both as genetic material and catalyst. D. Enzymes preceded genetic systems. Answer. C. RNA served both as genetic material and catalyst. CSIR Net June 2019. Which of the following features would most likely have been present in the first true cell? A. Nucleus and mitochondria. B. Double-stranded DNA and ribosome. C. Photosystems and chloroplasts. D. Multicellularity. Answer. B. Double-stranded DNA and ribosomes. Would you like the next advanced lecture on evolution of prokaryotes and eukaryotes, endosymbiotic theory, or RNA world in depth? All are closely related and frequently tested in CSIR Net JRF Life Sciences. You said. Evolution of prokaryotes. ChatGPT said. Here is your CSIR Net JRF Life Sciences ultra detailed lecture on evolution of prokaryotes, written with maximum word limit, in dense paragraph format, without tables, and designed to match the CSIR Net exam pattern based on previously asked question areas. This is your complete coaching material, crafted to help you crack the exam without needing other references. Evolution of prokaryotes, the origin and diversification of Earth's first life forms. The evolution of prokaryotes represents the earliest chapter in the history of life on Earth, beginning around 3.8 to 4 billion years ago. Prokaryotes were the first cellular life forms, arising from self-replicating molecular systems encapsulated in lipid membranes. These primitive life forms lacked a nucleus and membrane-bound organelles, characteristics that still define modern bacteria and archaea. The study of prokaryotic evolution is critical for understanding the origin of life, early metabolic pathways, and the biochemical foundation upon which all other life evolved. The earliest prokaryotes likely originated from protocells, self-assembled lipid vesicles encapsulating catalytic RNA and other molecules. With the advent of genomic stability through DNA, coupled with RNA-based catalysis and rudimentary ribosomes, the first true prokaryotic cells were formed. These organisms were most likely anaerobic heterotrophs, relying on environmental organic molecules for energy and carbon, given the absence of atmospheric oxygen in early Earth conditions. The metabolic capabilities of early prokaryotes were shaped by natural selection acting on molecular variation, leading to